Welcome to Click for IB Biology. We're studying C 3.1, Integration of Body Systems, and we're moving on to look at the integration of plant systems. We'll be studying the role of auxin and cytokinin. There's a PDF of this presentation that's linked into the description below the video, and also you'll find there connected, there are 10 questions, short answer questions, and the answers I've provided, and you can use those to evaluate your own learning. So the first aspect of plant integration of systems we're going to look at is the phenomena of tropisms, which are directional growth in response to an external stimuli. And in particular, we're going to be looking at the concept of positive phototropism. So this is positive, indicates that the plant is growing towards light. There's a unidirectional show of light here, and the plant is going towards that. So photo and then tropism, the directional growth. The phytohormones for this particular part are the auxins, and in particular, indole 3-acetic acid is the active phytohormone. Let's take a look at this. Now, phototropism has been documented since 1880, and indeed it was Darwin who did some of the early work in which he planted wheat seeds, germinated those into what are called Coleoptiles, the early shoots, and he noted that with unilateral light, the coleoptile tips grew towards the light. Darwin also took the liberty of removing the tip of the coleoptile, and when this happened, there was no unidirectional growth, there was no phototropic response. Another researcher placed small caps over the tip of the shoot, and again what resulted is that the shoot continued in this onward direction like so. To confirm that it's actually the tip of the shoot which is the sensitive part to light, the coleoptile tips were actually buried in sand like so, with only the tip exposed like this, and once more with unilateral light the shoot would grow towards that light in a positive phototropic response. Later on in the research through time, the substance identified as being active in this was called indole 3-acetic auxin, which is also known as auxin. So we shall call it that from now on. Later studies, by placing auxin on the coleoptile tips, like so in agar blocks, determined that as the concentration of auxin increased, then the degree of bending would also increase. This is a fairly complex relationship, but let's move on to what is now established as the action of auxin. So what we're trying to explain is that with unilateral light coming from this side, there is growth towards the light. And this growth occurs through the elongation of cells on the what I will call the shaded side of the plant. There is asymmetrical growth, and in this what's happening is that the cells in this area here, the cells are elongating, whereas on this side the cells are still small like so forth, and so we get a bending action towards the light. It's believed, and it was hypothesized, that this was due to a lateral transport of auxin. The mechanism would be later revealed. The lateral transport of auxin is understood to be due to, first of all, the production of auxin from one cell to another. So if we take our coleoptile tip like so, there's the downward movement of auxin from one cell through the top of the cell through what are called the aux transporters. And these are at the apical surface of a cell. Now, ordinarily, what would happen without the strong light from the side is that the auxin would then be transported downward by transporter proteins called pin proteins or pin transporters. However, with strong light, what happens is that the pin transporters are relocated to the lateral membranes, and the auxin is moved towards the shaded side. The effect of auxin is to, in the cell wall of the plant, if I show it like so, the effect of auxin is to release the cross linkages between the cellulose fibers so that the wall becomes flexible. Water entering the cell causes expansion by osmosis, and the cell effectively stretches to a longer length before the cellulose in the wall reforms the cross linkages, securing the stable wall once more. In this way, we have asymmetrical growth on one side of the coleoptile tip. Auxin has a second function. Like many regulatory molecules, auxin synthesis is known to take place in the apical meristem, the growth point of the shoot. And we've already discussed the pin transport downward. Now, it turns out that the second effect of auxin is what is called apical dominance. And we can see that here in the apices here, in the angle here between the stem and the petiole of the leaf, there is what is called a lateral bud. And in effect, the growth of the lateral bud creates a branch. Auxin, transporting downward from pin to aux to pin, moving down, inhibits the development of lateral buds. The transportation of it is also involved in the development of phloem transport. And so we can see in the diagram here that the production of the auxin at the top would have the highest concentration, and then auxin concentration will decrease as it goes down the stem, reducing its influence. And of course, this reminds us that where we are affecting the growth of an organism through gradients, suggests that auxin is what we would call a morphogen. At this point, we should also consider the second phytohormone known as cytokinin. Cytokinin is produced at the opposite end of the plant in what is known as the apical meristem of the root. And at the apical meristem of the root, cytokinin is responsible for the development of root tissue. The role of cytokinin in the roots is to inhibit the lateral development of roots here and maintain the forward growth of the roots down into the soil. Of course, we have two contrasting gradients here, with cytokinin also acting as a morphogen. So auxin inhibits lateral shoots and cytokinin inhibits lateral roots. The ratio of auxin to cytokinin determines whether or not we have lateral shoots or lateral roots. If we have a high concentration of cytokinin relative to auxin, then we will get the development of lateral shoots. They will grow outwards like so. If, however, we have where the concentration of auxin becomes high, in the root system, this encourages the development of lateral roots. Of course, that would occur in a more mature plant. Interestingly, in gardening, if one removes the apical meristem and the origin of auxin, what happens is that the plant will develop many lateral branches and become more branching, which for some gardeners is seen as a desirable characteristic. In this way, auxin and cytokinin combine their effects and integrate the growth of the plant through their morphogenic effects and the development of the tissues. Link to the next video on fruit ripening and ethylene gas is on the left-hand side. A chance to subscribe to the channel is in the center, and the playlist is on the right-hand side. Thank you for listening to this long video.